everyone. Thank you for being here. It's a real honor to be able to present here and tell you a little bit more about our company. It's Pet Bio. We're based in this beautiful building in West London in the UK. We're going to talk about mainly our approach to precision medicine in oncology and the role that computer vision plays in that approach. So these points have been made by many people throughout this conference, the sort of problems with current cancer models, so I won't belabor the point. For us, the main issues is that the tumor microenvironment is not replicated, and the immune response is also generally lacking. In the case of patient-derived xenografts, the studies are also too lengthy in order to direct the treatment of the patient, which is our ultimate goal. So there's a need for functional models with readouts that clinicians can also understand and that can direct the treatment of the patient in a reasonable time frame. So this is sort of our proposed sort of engine. Every patient is unique, and therefore their treatment decisions should be treated as such. In order to do this, we need to use their own cells, and we need to replicate the tumor microenvironment as best as we can. So in order to test the sort of available drugs, we make patient-derived microtumors, and we test them against a panel of both combination and monotherapies that we would reasonably expect they could receive in the clinic. So we don't sort of stratify or test or anything like that. We try and test anything that they might be able to get because we're testing while they themselves are sort of being evaluated for their treatment. And then we hope to basically be able to predict the tumor response or the patient's response even to one or several treatments by doing daily live confocal imaging, and we provide a report using computer vision analysis of those images, and the hope is that we can sort of provide guidance on complex indications, for example, triple-negative breast cancer. So as I mentioned, it all starts with the primary tissue. This is sort of an overview of our workflow, both sort of our prospective workflow in a clinical context and also our workflow in the development of the model. We're working very hard to get as many viable cells out of each sample. Each cell is precious to us, and we really expect that having as many cells as possible means we can get as accurate information as possible. We get the patient tumor as well as blood fraction, which we can isolate PBMCs from. We dye the tumors one color, the PBMCs another color, and we can incorporate those populations within a hydrogel. So we actually tailor the hydrogel to each to each tumor type. I'm a biomaterials engineer in the company, so it's a big part of my day-to-day. And we do that by testing many formulations. Currently, I'm testing formulations for renal cell carcinoma. I've tested over 50 formulations of hydrogel. But the starting point is kind of always the literature and finding out about the tumor matrixome for those specific cancers. Once we've set up these 3D cultures, we image them daily using a confocal microscope, as I mentioned. We have 3 confocal microscopes in-house, which I think is pretty rare for a startup of our size. And we take that confocal data, 3-dimensional confocal data, and send it to our in-house computer vision team. So we have a couple of software engineers who are developing basically ways of analyzing this imagery data and pulling out as many metrics as possible. These could be things like cell viability, cell migration, cell position changes over time, cell size, and also the volume change of the microtumor itself. At the end, we hope to basically put all of this information together in a report, which is easily readable by an oncologist and direct the treatment. The important thing as well is that this is done within sort of a week, because for a lot of these cancer patients, they don't have a lot of time to wait around and get given treatment. So we want to really be able to direct their treatment as quickly as possible. So these are a sort of a summary of all of the cancers that we've been able to test so far. So we've tested, at this point, actually over 220 patients mainly in breast and within breast, mainly triple-negative breast cancer. As I mentioned, there's a lot of ongoing development right now in kidney cancer, which is most of those cases, as it is in the clinic, have been clear cell RCC. But we're also actively looking at liver cancer, hepatocellular carcinoma, and PDAC. So this is just an overview of a few of the different architectures that we generate. So these are all gel domes within a 96-well plate or a microfluidic chip. You can see the heterogeneity of the different organs. They can look quite different between, say, breast cancer and kidney cancer, for example. This kind of just gives you an idea of the density as well that we are generally trying to achieve in each of our cultures. These are about between 5 and 10 microliters each, generally 5 to 10,000 cells in each as well. So now I'm going to go through a sort of real-world example of an RCC culture we tested with matched PBMCs. So the way we add the PBMCs sort of currently is on top of the gel dome in solution. We can either sort of add immunotherapies alongside the PBMCs in the media, or we can sort of prime the PBMCs by adding immunotherapy to them in suspension shortly before adding them to the cancer cells. Generally, we dyed the tumor cells yellow, the CD8 negative PBMC fraction red, and the CD8 positive T cells blue, and then we've just got enough sort of spectral room left to image dead cell stain in the green channel. Generally, we don't find that there's too much crosstalk in between these channels. We've got really high, state-of-the-art confocals, and it's something that we're very aware of. So our computer vision processing looks at these individual channels and can find, the tumor cells within the dome, the PBMCs outside, and the ones that have infiltrated inside the dome as well. There's dead cells, and I'll talk a bit more about that in a second. We can also look at sort of more time lapses of our cultures, so hopefully that video plays. We can see the CD8 positive T cells infiltrating from the side of the dome. Inside, we look at the whole dome, really the whole shape, so we see all of the PBMCs invading from every side. This is with and without Keytruda, which is a known immunotherapy. So the computer vision, it'll detect these single cells across all of the time points. So what we're getting here is the spatial positioning of each cell, the size of each cell, and we can use the tumor cells themselves to generate a basically map of the dome of the microtumor, and then we can use that to basically define whether or not the immune cells have invaded into that dome. With the dead stain, we can also basically measure the degree to which the dead stain and the tumor cells or the CD8 positive T cells match, so looking at overlap, and we can use that overlap as well to see points in time where CD8 positive T cells might be right next to tumor cells. Looking in a bit more on a much more high resolution, temporal resolution, you can see here a short video where we saw CD8 positive T cells attacking a tumor cell, which is in yellow, alongside other PBMCs, and eventually the tumor cell is basically, its membrane is compromised, and the dead stain can go in and bind to its DNA, and then it shows up green in our test. So our hope is that we can really use this sort of these 3-4 colors to measure these events over time and form a basis of the predictive value of our test. In terms of actual metrics that we get, so this is early data from a single patient, but it's just an example readout of the types of things we'd like to measure. So it could be CD8 positive invaded numbers with and without a drug. It could also be change in the live tumor cells over time. So you can see here we're doing this test sort of over 4 days. We hope to be able to get as much predictive value out of that short time scale as possible. But before we can put this in the patient, or sorry, not in the patient, before we can put this in the clinic, we need to develop these models. So we need to validate them once a protocol or method shows promise, and sort of we need to understand the underlying biology before we can really direct patients' treatment. So a big part of what I have to do is develop the hydrogels for these different cancer indications. This is sort of an example from my recent 3D RCC model development, and these are the types of assays that I use to really understand, are the cells metabolically active? Are they secreting soluble factors? Is the population of cells sort of similar to what you would expect from the native tissue or from the native tumor? So we look at, like I said, metabolic activity over time. Nan, who's in the audience, did some great work in immunofluorescence looking at different cell populations. So you can see here that we've conserved at least several different cell types, so cancer cells, endothelial cells, and fibroblasts. We don't do any pre-sorting of the cancer tissue at all. We just take everything that's in there. We can also look at live dead. This slide is actually more to show the difference between the different formulations and how live dead or viability is not the only metric that you need to sort of examine. Here, these different formulations, some are based on collagen, some may contain matrigel, fibronectin, and you can see that our two leading formulations at the moment are RENA F33 and RENA F40, and they have very similar viabilities at this sort of final time point. But the PBMC's infiltration, even though this is on a different patient, it's a trend that we've seen repeated across several patients, is very different. So is it something about the structure of the hydrogel that means that PBMCs are able to infiltrate more easily in RENA F33 than RENA F40, which is the red line? Is it something about the signaling that actually for the PBMCs that means that they invade more easily? We're not entirely sure yet. This is something that we're sort of, alongside all of the other assays that we do, are trying to sort of figure out. So this next slide is sort of about our goals in predicting patient response. So this is in triple-negative breast cancer. So this is sort of early results, again, from our first observational clinical trial, which is ongoing. So this was looking at 4 patients and looking at a response in a neoadjuvant context. So we can see here that some of our metrics that we're able to pull from the computer vision, such as relative cell viability or a change in live cells, that's kind of comes from the same number, really. It's just different ways of presenting the same data, as well as the tumor core diameter, which is also measured by the computer vision, show sort of nice predictive of pathological complete response versus non-complete response. So about 6 months after the patients start their neoadjuvant treatment, we get that data back to us to understand if they've completely responded or not. And for each metric, we basically set a numerical threshold and analyze the patient population into either predicted PCR or predicted non-PCR. So that could be, we say, for example, if the cell viability is less than 50% on the last day, then the device is considered to be sort of predicting complete response. We use an algorithm to tabulate the predicted response against the real-world outcomes that we get back, and these sort of specificity and sensitivity values for the threshold are plotted on what's called a receiver operating characteristic curve, which you can see 2 examples there for 2 different metrics. And in order to be considered an indicator of pathological complete response or non-response, the corresponding ROC curve must meet at least 80% specificity while retaining adequate sensitivity, calculated by the partial area under the curve of being over 70%. So this is kind of our ongoing trials. As you can see, we've kind of moved quite forward in quite a few of these now, especially in PAIR-TNBC and PAIR-MET, so that's going to be looking at metastases of TNBC. In the kidney, we're sort of almost done optimizing the analytical workflow, so we'll be looking at retrospective testing as well. In pair PAL in the liver, we're looking at optimizing the workflow, and that's in partnership with Innovate UK. And pair Glio in the brain is sort of in the early stage of R&D. Our partner hospitals are also there on the slide across London. So with that, I'd like to thank all of the team at Pair Bio. Ellie and Nan are here as well, so you can speak to them and me if you have any more questions. And especially I'd like to thank all of the patients, the oncologists, the pathologists, and our hospital and biobank partners for allowing us to use their tissue to develop our technology. As Sheena said, we couldn't do it without them, so thank you very much.
Questions for Thomas?
Thank you. Really nice work. I'm curious, I mean, so of course one of the challenges that you have is that you are running against time, right? I mean, the patient is waiting. So one of the challenges that you face in this model is the fact that the patient is waiting, and so you only have so many days to produce a result. So, I mean, in instances like these, when you're talking about stimulation of immune infiltration of the tumor tissue, I think it's reasonably, I'm not going to say easy. I mean, it's easy to understand how you ascertain efficacy. But I mean, if you're dealing with a drug that whose mechanism is, say, cytotoxicity, something like that, does that bring more of a challenge given the short time frame that you have in your hands to come up with it?
Yeah, so that's a great question. I think there's definitely limitations, obviously, to trying to test these things so quickly. The hope is that we can still get, be able to predict response, even though biologically, compared to what might be done in the clinic, it's not necessarily super representative. You might have noticed vasculature is missing from a lot of these cultures because we don't want to introduce UVEX. We want to keep it as patient-centric as possible. With that, we're just really trying to get the simplest but still predictive model that we can.